Researchers net $85 million to tap ancient eDNA to inspire next-generation climate-resilient crops. The early data is breathtaking. June 13th, 2024, Elaine Watson. A pioneering project exploring ancient environmental DNA, eDNA, genetic material left by plants, animals, and microbes in soil, sediment, ice, and other sources up to 2 million years old, could help scientists develop more climate-resistant crops by understanding how ecosystems have adapted to environmental change in the past. The Ancient Environmental Genomics Initiative for Sustainability (AEGIS) project has been awarded Dutch kroner 585 million, which is 85 million US over 7 years, with the lion's share 72 million coming from Danish nonprofit Novo Nordisk Foundation and the remainder from UK charity the Wellcome Trust. By sequencing DNA fragments from samples all over the world and comparing them to the DNA in modern species and reference genomes, researchers can better understand past genetic diversity and learn how ancient species adapted to their environments, claims research leader, evolutionary geneticist Professor Eske Willerslev from the University of Copenhagen and University of Cambridge. While modern food crops are typically bred for traits such as higher yield or disease resistance, their wild-type ancestors had far greater genetic diversity, developing a wide range of genetic variations that help them survive in diverse and changing conditions, he said. By employing ecosystem modeling, we can pinpoint which combinations of species led to the most durable ecosystems in the past. This knowledge could serve as a blueprint for creating climate-resilient food systems, enhancing both the crops we grow and the sustainability of the environments they grow in. AGIS aims to gain insights from ancient eDNA to understand ancient genetic diversity in plants, identify past climate adaptations, and apply these findings to modern crop breeding, including barley, wheat, and rice. We had the Human Genome Project. This is the Planetary Genome Project. The expectation is that AEGIS will demonstrate proof of concept in key crops such as barley, wheat, and rice, say the researchers, who span disciplines including bioinformatics, AI and machine learning, microbial ecology, plant breeding, and environmental genomics. They will also create a public data portal featuring climate data, time points, sample types, genomic data, and reference genomes collected during the project, Novo Nordisk Foundation SVP Dr. Klaus Felby told AgFunder News. There's DNA everywhere. In a teaspoon of cell, you can see DNA fragments from everything from bacteria and fungi up to the biggest mammal. So we had the Human Genome Project. This is the Planetary Genome Project. You can map everything in a given ecosystem. Every plant, tree, microorganism, every mammal that was there at a given point in time and then over time. So you can see how the ecosystem evolved and adapted over time. I can say that initial results so far are breathtaking. So how far back are researchers looking? Anything between 10 to 15,000 years, that's highly feasible, said Felby. And if you're lucky, you can go back much further. I think the record for collecting usable DNA is about 2.5 million years. He added, I can say the initial results so far are breathtaking. For instance, we have found how rice adapted to a much wetter climate in a part of China about 8,000 years ago. And you can see down to the single gene how the whole ecosystem adapted. You can see how the microbiology of the soil has changed. According to Felby, you can read the climate at the same time as in analyzing DNA fragment from a given period by looking at things like oxygen isotopes, because they change according to precipitation, temperature, and so on. So then you can start to map the two things. But we're not just looking at rice or any given plant, we're looking at the entire ecosystem, and if you see changes in all the plants, there's a pattern. Bioinformatics big time. Stepping back, he said, we often speculate about what's going to happen to our ecosystems with climate change, but all these experiments have been done before if you look back far enough. And now we can read the results, and that's fascinating. But it also allows for much more targeted and much faster plant breeding if you know where to look. It allows for polygenic traits, not just a single gene, but you see multiple genes changing at the same time. It's like if you suddenly got a spaceship that would allow you to fly from one end of the solar system to the other in 2 hours. And now we have AI and machine learning and super fast computers that we can handle these vast data sets. It's bioinformatics big time. So as part of this project, we are also building one of the world's most powerful supercomputers here in Denmark with NVIDIA. Metagenomics, the study of genetic material recovered directly from environmental samples, has been around for a while, he said, but this is the first time we are doing it at full ecosystem scale. We can map not just the effect of climate and climate change, but also what happens to an ecosystem when humans enter. What happens when we develop agriculture? What happens when we start to apply fertilizers and pesticides? According to the European Molecular Biology Laboratory's European Bioinformatics Institute, based at the Wellcome Genome Campus near Cambridge, United Kingdom, through human cultivation, crop plants have lost the genetic diversity that was present in their wild-type ancestors. Ancient environmental DNA found in the soil can help researchers understand how the ancestors of crop plants adapted to historical climate change. These insights can be used to develop new strategies to make modern crops more resilient and provide novel solutions as climate change continues to threaten global food security. End of article. So this is an interesting article, and it mentions here that the two companies that are funding it are the um, Danish nonprofit Novo Nordisk Foundation. That's $85 million over 7 years, and then some of that money, um, or 13 million, is the United Kingdom charity, the Wellcome Trust. So that's about, seems to be an 8-year funding runway to run the data. And of course, they're using NVIDIA, not surprising, and they're building one of the largest supercomputers in the world in Denmark in order to run and build this data set. One of the world's most powerful supercomputers in Denmark with the help of NVIDIA. Very interesting. So it'll be hopeful to see how fast this can be built and the data that can be rendered and yield from it, and how it will be applied back to various industry players in order to um, go back in time and understand the genetic resiliency or diversity of plants that were eventually sort of domesticated and farmed at scale, such as wheat and other plants, as they've mentioned in this article. So it should be good to know that there's other projects not mentioned in this article that are doing the same thing, is that they are going to archaeology sites and they're going to museums of all places to take samples of wheat and barley and rice and whatever that have been lying dormant for thousands of years, in order to also tap into the genetic uh, history and crossbreed and yield the traits of those seeds, because those seeds have been essentially in statu and not deployed for thousands of years, just lying there for later deployment. This project hopefully will be able to also go back in time and find all of the genetic sequencing of plants, and then maybe it will be able to provide this information and data set, and then people and researchers with the ability to deploy or tap into certain traits and genes can create super crops of a whole nother scale. That would be ideally the most commercial and viable way and high yielding way to take advantage of this project. Okay,